Sige. Dun sa mga natapos na natin kanina na subtopics, may gusto ba kayong ipaulit? Or are we okay na there? Okay na. Sige, continue na natin para maaga na din tayong matapos. Saka para makapag-lunch din kayo ng mabuti. So, for that of the plasmids, again, this would actually be tackled more during bacterial reproduction. So, plasmids, these are extra-chromosomal double-stranded element of the DNA that is associated with virulence. Again, when you say virulence, it's the ability of a particular organism to cause that of a disease. So, it's located in the cytoplasm and it will serve as a site for the genes to code for antibiotic resistance and toxin production. Like I was saying before with regards to MRSA, that is resistant Staph aureus, most of the mutated genes or those genes that would actually carry antibiotic resistance, they would be incorporated in your plasmids. So it's not essential for bacterial growth. So a bacterial cell may or may not contain a plasmid, but it would actually sometimes disappear during cellular division, and it can make bacteria, mostly of yung ating gram-negative, pathogenic because yun na nga, it would actually be associated with that of your own A in trying to contribute to that of resistance. So not required for growth and reproduction, but it will carry genes that is needed for drug resistance, pathogenicity, and for new metabolic activities. So the two kinds of plasmids, we have the large and the small one. So the large plasmid is responsible to produce your beta-lactamase that will provide resistance to beta-lactam antibiotics. The beta-lactam antibiotics like your penicillin, oxacillin, they have that particular structure, yung beta-lactam. And since the plasmid would have the enzyme beta-lactamase, it tends to cleave yung beta-lactam, rendering those antibiotics ineffective. So magiging resistant yung mga organisms. So small plasmids naman, they are the ones that are resistant to tetracycline and chloramphenicol. So that's basically for plasmid. Eventually, mas maiintindihan natin yung function niya kapag nandun na tayo sa bacterial reproduction. So for that of inclusion bodies, ito yung parang reservoir ng mga nutrients. So it would actually serve as the energy source or food reserve of the bacteria or as a reservoir of structural building blocks. So it's actually composed mainly of your polysaccharides and they tend to lessen the osmotic pressure. For inclusion bodies, they usually appear as refractile bodies that are suspended in that of the cytoplasm once you would actually be using that of your phase contrast microscope. And most of yung ating inclusion bodies, they are bounded by that of a thin membrane. Usually, this membrane of theirs would actually be composed of lipids. And the lipid present in that of the membrane of this inclusion bodies will tend to separate that of the inclusion bodies from that of the cytoplasm. So the inclusion bodies, these are granules of organic, inorganic materials that are again needed for the different metabolic processes of this bacteria. So the two most common types of granules, masyado kasing marami talagang inclusion bodies. But the two most common would actually be your glycogen and that of your polyphosphate granules. So glycogen, as you all know, even in that of biochemistry, it would actually be the storage form of glucose. And glucose is actually necessary in order to make ATP, in order rather to provide energy to that of the bacteria. And then we have polyphosphate granules, or according to Jowett, it's actually termed as volutin or metachromatic granules. So yung polyphosphate, with the word phosphate, so it would actually be the storage area for that of the inorganic phosphates. And phosphates, as we all know, it's very important in making that of your nucleic acid for that of the DNA, and that of also for phospholipid synthesis for that of the membrane. Kapag sinasabi kasing metachromatic granule, when you say metachromatic, it will present a different color that is expected of that particular stain. Like for example, if you would actually be using a blue dye, you expected, di ba, dapat na color would actually be color blue. But instead of color blue yung nagpakita, the organism would actually then appear red. Kaya metachromatic. It's actually of a different hue, a different color than what is expected. So, an example would actually be your Babe's urns bodies that is very much characteristic for that of your diphtheria. So this is actually an example of your Babe's urns. So yung mga highly concentrated na areas dun sa dulo, instead of them being stained as red, they would actually appear as blue or dark blue. So those are actually the polyphosphate or metachromatic granules of your diphtheriae. And then another very important example are the bipolar bodies of your Yersinia pestis. Yersinia pestis, which is the causative agent of the bubonic plague or the Black Death during the Middle Ages. So yung bipolar bodies, kaya siya tinawag na bipolar bodies because at the end of the said bacteria, yung sa magkabilang dulo, nandoon yung ating bipolar bodies. So it would actually contribute to that of the safety pin appearance ng ating Yersinia pestis. So pag may narinig na kayang safety pin appearance, huwag na mag-iisip ng iba, Yersinia pestis yan. And that's actually because of your bipolar bonds. And then we have your muck granules. Ito naman, it would actually appear as pearls, pearl-like in appearance. That's actually associated with your Mycobacterium tuberculosis. So next, we have your polybeta hydroxybutyric acid. It's a lipid-like compound consisting of chains of your beta-hydroxybutyric acid that is actually connected through ester linkages. So it's actually produced when the source of nitrogen, sulfur, or phosphorus is limited. And at the same time, there would actually be increasing carbon in that of the medium. Again, there are certain microorganisms that would actually have very unique requirements for that of its growth. Some would actually need sulfur, some would actually need that of phosphorus. But if these particular substances would actually be limited in that of their environment, they tend to shift to another substance in order for them to continue on living in that particular environment. So for PHB and glycogen, they are also carbon sources when protein and nucleic acid synthesis would actually be resumed. And sulfur granules, they would also be packed into inclusion bodies in the form of hydrogen sulfide and thiosulfate. There are certain microorganisms kasi that would actually then require sulfur. So any questions so far? Wala nang katanungan?
Wala na po.
Okay. So for that of the endospore, it's actually the toughest of all specialized cells. But again, not all organisms, not all bacteria are capable of forming spores. They are longest surviving if they would actually be capable of undergoing sporulation. And itong mga endospores, they may remain dormant, inactive for many periods of time. Kaya nga, di ba, for those particular agricultural lands, especially here in the country, who have actually experienced anthrax before, yung anthrax kasi na bacillus, it is actually capable of forming endospores. And sometimes, they would actually remain unnoticed. They tend to be embedded in that of the soil. Kaya nagiging dormant, inactive sila, and for such a long period of time, they can withstand any type of environmental condition. Kaya nga, kahit ilang years nang natapos na nagkaroon ng anthrax outbreak in that particular region, in that particular area, pwede ulit mag-recur because of certain spores that are embedded in that of the soil. So, for endospores, they would actually survive treatment that will rapidly and efficiently kill the other cell forms. So, kahit mataas na temperature, then they can actually withstand that even with radiation, detergents, or any other type of hydrolytic enzymes. So again, the two most important bacteria capable of doing endospores would be the gram-positive anaerobic bacteria, your Clostridium, and the gram-positive aerobic bacteria, your Bacillus. So again, they are small dormant structures located inside that of the bacterial cell. They will aid in survival of the bacteria against external conditions. Kapag nasisense kasi ng bacteria na limited na yung nutrients in that of its surrounding, or there is actually a threat to that of its survival, it would actually initiate that of the sporulation process. So the endospore, it's actually produced within vegetative cells of some gram-positive bacteria. I know you're well aware na of the term vegetative kasi di ba na encounter nyo to sa parasitology. When a particular organism will tend to feed that of itself. Kapag kakain siya, that is actually what you call the vegetative state. But in that of endospores kasi, if it's actually limited, yung bacteria na sa sense niya, limited yung nutrients in that of its environment, it can go into sporulation. But once na-sense niya ulit na there would be abundance of different nutrients for that of its survival, it would actually then shift into that of its feeding state or its vegetative state. So the endospore is a very tough structure to crack because it's actually composed of dipicolinic acid and your calcium ions. It's collectively termed as your calcium dipicolinate. Kaya it's very much resistant even to that of the harshest environmental conditions that are present. So some locations of these spores, it can microscopically identify the bacteria. Like, for example, here, kapag nandoon siya sa dulo, that's what we call as the terminal spore, nandoon siya sa pinakadulo, that is a terminal spore and it is actually characteristic of your Clostridium tetani, which is actually the causative agent of your tetanus. And then we have the subterminal naman natin na spore. Makikita nyo, di ba, hindi siya mismong dun sa dulo talaga, but it's actually near that of the edge. And that is your very much characteristic of your Clostridium botulinum. Clostridium botulinum, which is actually a major source of food poisoning, commonly called as your canned food or canned good bacillus. Kaya nga, di ba, yung mga canned goods na hindi talaga na-store properly, it can actually be a cultivating ground for that of Clostridium botulinum. Kaya nga, di ba, sinasabi lang kapag doon pa lang sa mismong lata, it's not in very good condition na, alam mong hindi maganda yung pagkaka-transport, yung pagkaka-store, huwag mo na lang kainin. Kasi it can actually cause food poisoning. And then we also have the central spore that's very much characteristic for your Bacillus anthracis. So central spore, nandoon siya sa mismong gitna ng Bacillus. So again, that's an example for your Bacillus anthracis for anthrax. At kung makikita nyo nga dito, di ba, para siyang chain yung kanyang arrangement. Kaya nga yung Bacillus anthracis natin, it's sometimes being described as to having a bamboo appearance. Kasi para siyang bamboo. So for that of the spores, they are responsible for perpetuation but not multiplication. And the transformation of the endospores to that of the vegetative na din na form, it would actually require a series of steps din. If you have actually read na in advance sa inyong jawets, na-discuss niya to na mabuti. So, we have your activation, the germination, or the initiation phase. That is actually associated with activating your autolysin and outgrowth. So ano bang nangyayari sa ating activation? Initially, in that of activation, the membrane or that of the coat in that of yung ating spores, they are being triggered by that of the amount of nutrients, substances in that of its environment. So anong ginagawa niya? Kapag sa tingin niya ng madami ng nutrients sa kanyang environment, it tends to lyse, to damage that of the coat, the spore coat. So kapag na-damage niya yung spore coat, ang ultimately lalabas is that of yung ating membrane, which is actually composed of your peptidoglycan. So, on the in that of yung ating initiation stage, certain effectors would actually act as a signal. These are actually present in that of the environment, and they would actually act as a signal to activate the autolysin that eventually degrades that of the peptidoglycan. So natanggal na yung coat, and then sa initiation, natanggal pa yung ating peptidoglycan. So wala nang barrier. That is actually in order to get all of the different nutrients in that of the environment. Kasi for such a long time, it actually reserve yung remaining nilang nutrients. So ngayon na marami ng nutrients in that of the environment, it will open that of its barrier and take all of the different things that it will need for it to survive. So, yun yung nangyayari sa ating initiation. And then, upon getting all of those different nutrients, mag-i-start na siya with the different metabolic processes to create the macromolecules that is needed for that of its growth. And then, magkakaroon tayo ng cellular division. And the cellular division itself, that is what you call as the outgrowth. So, dadami siya. Paano naman nagagawa yung sa ating endospore? So, in that of a harsh environment, or in that of an environment to which the needed na organic and inorganic substances would actually be limited, the growth of the organism would actually stop, and the DNA would then be duplicated. And then a septum will then be formed in the middle that will then divide the cell asymmetrically. Kaya magkakaroon siya ng large at saka smaller compartment. And depending on that of its need, it can actually further engulf yung large component, i-engulf niya yung smaller compartment. Depende kung gaano pa kadami yung kinakailangan niya at such a limited condition. And then, peptidoglycan containing material will then be laid down between the two membranes so that it actually forms a barrier na naman. And then eventually the mother cell will then be degraded, and then the endospore will be released. So it would actually be a very complicated process in order to protect itself from that of the environment. So any questions so far? So for that of the different structures of the endospore, again we have the core, which is actually being regarded as the spore protoplasm, containing all of the different structures, the complete nucleus, all the components of the ribosome, and an energy-generating system based on glycolysis. We also have the spore wall, or the core wall, the innermost layer surrounding the inner spore membrane, and it is the one that would actually contain the normal peptidoglycan layer and becomes the cell wall of the germinating vegetative cell. And then the cortex, which is the thickest layer, it contains an unusual type of peptidoglycan because it would actually have fewer cross-links than those that would actually be found with the other organisms with peptidoglycan. And yung ating cortex is extremely sensitive to that of lysozyme. Miss Machas?
Yes,
Miss
ma'am.
Machas, nandiyan ka pa ba? Miss Machas, what are the 3 different body fluids that you have actually encountered in your anatomy that is very rich in the enzyme lysozyme?
Saliva, ma'am.
Ano pa? Tatlo yun eh, di ba?
Saliva.
So the 3 different... Sige, thank you. So yung 3 different body fluids that are rich in lysozyme would be your tears, your saliva, and that of also breast milk. Kaya makikita ninyo, initial defenses pa lang of the body would actually be effective in trying to prevent the colonization of spore-forming bacteria. So meron tayong defense mechanism against spores. Next, we have your spore coat that's actually composed of keratin-like protein, like the keratin that's actually present in your hair, in that of your nails, in that of your skin. And it would actually contain many intramolecular disulfide bonds. And the impermeability of this layer of yung ating spore coat confers on spores their relative resistance to antibacterial chemical agents. Again, all attributed to that of yung ating spore coat. And then we have the exosporium, composed of proteins, lipids, and carbohydrates, consisting of a paracrystalline basal layer and a hair-like outer region, and it is seen in your Bacillus species, for your Bacillus anthracis and your Bacillus cereus. Bacillus cereus, which is also another causative agent for food poisoning, and it is commonly known as your fried rice bacillus. Kaya nga, di ba, kapag yung fried rice, you would actually just leave it there sa table, and then without actually any types of preservatives that were actually added, it can actually be, again, a cultivating ground for that of your microorganism, for that of your Bacillus cereus. So it will also cause that of your food poisoning. So the other structures external to that of the envelope, so we have the cellular appendages. They would play a role in the mediation of infection, and in the laboratory, it would actually be needed for identifying that of the organism. It is different between species and even among strains within the same species. And one of which will have yung ating glycocalyx, the umbrella term for that of your capsule and your slime layer. So see, glycocalyx, it's an outward complex of polysaccharide on the bacterial surface and the other cells. It will help the bacteria attach to surfaces, either animate or inanimate surfaces. And again, it would actually be present as either a capsule or that of a slime layer. Ano mang pagkakaiba kapag capsule dun sa ating slime layer? A capsule is a condensed, well-defined layer closely surrounding the cell, and it tends to exclude that of the other particle. So it's firmly adherent to that of the cell, excluding the other particles. An example that was actually given in your book was the India ink particle. India ink is actually a stain that is a type of negative staining. When you say negative staining, instead na yung microorganism ang ini-stain niya, yung background ang ini-stain niya. That's actually because of the capsule. Sabi nga natin, di ba, it excludes the particles. It will never take the stain. It will never take the dye. So instead of coloring that of the organism, pupunta na lang siya dun sa surrounding ng organism. So ang may stain, yung background. So that the organism will then appear to have a halo or a clear region around it against a black or dark background. That is actually for that of your engine staining and that of also your capsule. Paano naman yung slime layer? The slime layer, as we have actually discussed dun sa opening, that it is loosely attached, loosely adherent to that of the cell wall, and it does not exclude the particles. It would just take in any other particle that would actually be present in that of its surrounding. That is your slime layer. So the glycocalyx in general is a gelatinous polymer extending from the surface of the bacteria to sometimes involve the other cells. And these other cells, it may actually be another type of yung ating bacteria, either of the same species or of a different group. And the importance of the glycocalyx would be protection against ion and pH changes and osmotic stress. Again, protection against certain enzymes, especially if it would actually be present in the form of a biofilm. Protection against predaceous, opportunistic bacteria and phagocytic cells like your neutrophils and your macrophages. And all in all, it enhances the virulence of the pathogenic bacteria kaya hindi sila madali mamatay. So again, for capsule and the slime layer. So the capsule, the layer is well organized. It's firmly adherent to that of the cell wall. It is not easily washed off kasi sobrang nakadikit siya. Sa slime layer, it's unorganized, it's diffused, and easily removed because, sabi nga natin, it's loosely adherent, loosely attached to that of the cell wall. So going further to that of the capsule, again, paulit-ulit, organized, condensed, firmly attached to cell wall, immediately exterior to the murine layer of gram-positive and the outer membrane of your gram-negative. So it's actually made up of polysaccharide polymers except for your anthracis and your licheniformis, because it's actually composed of the poly-D-glutamic acid. So, kung makikita nyo dito, di ba, ito siya. It would actually indicate na mayroong capsule yung ating microorganism kapag mayroon kayong makikita parang halo appearance around the organism. And this is actually a poorly done India ink staining kasi dapat sana talagang black na black yung background niya. And then, maglalighten yung capsule around that of the organism. Maganda talagang tignan yung ating negative staining. So, for that of the capsule, it protects the bacteria, again, contributory to that of its virulence, from the attack of human defense system, since it would actually resist your phagocytosis, your drying or desiccation. And capsules sometimes must be removed to detect the somatic antigen that would actually be present. Again, if in laboratories kasi, they tend to do this. Actually, hindi sa the clinical laboratory, but in those laboratories that are for researches, they would actually try to remove certain parts of yung ating bacteria in order to make an effective antibiotic against it. And somatic antigens, for example, that is actually very common for that of your salmonella. And then capsule removal is accomplished by boiling a suspension of the microorganism in order to disintegrate, to destroy that of the capsule and eventually expose yung kanyang peptidoglycan layer. And it does not ordinarily stain, again, with the use of common lab stains. Kailangan yun nga ng specialized na technique. In that of performing India ink, which we will discuss sa laboratory soon, hindi din siya basta-basta staining procedure. So, but nonetheless, yung ating capsule will always appear as a clearer area, a halo-like area around that of the organism. So, for that of the slime layer, this is actually an example of the biofilm that is actually being caused by the slime layer of microorganisms. So, the slime layer is an unorganized material that is loosely attached to the cell wall. It's made up of polysaccharide. It can either inhibit phagocytosis or just be for adherence lang to that of tissues or foreign materials, yung mga implants. Like for example, yung nga, prosthetic heart valves, catheters, other na tubings na ini-insert sa katawan. So it facilitates and maintains bacterial colonization of biologic surfaces like in between your teeth, yung mga plaque, mga dental caries na tinatawag natin. Those particular organisms that are present in between that of your teeth, specifically the ones that are pathogenic, mga nakaka-cause ng disease, they have yung ating slime layer. So yung slime layer kasi na yan, it would actually be able to protect, para siyang shield, para siyang blanket, na hindi ng isang grupo ng bacteria, pwede iba't ibang types, iba't ibang species of microorganisms. And kaya nga yung ating mga dentists, they would always say that we need to brush our teeth rather regularly. Sabi pa nga, di ba, na most 3 times a day, that would actually, or every after you would actually eat, you need to brush your teeth. And you need to also do your flossing after you would actually brush your teeth. Kasi sometimes nakadikit sila in between, di ba, that of the teeth. And itong mga organisms kasi dun sa mga dental caries natin, they can be carried by that of the saliva. So that kapag lumunok tayo or during that of our sleep, and then the saliva would actually go to that of the pharynx, it can actually also cause neck infections. Kaya nga, most of the neck infections, they are being caused by dental caries. Or it can actually be yung slime layer natin. It can actually maintain bacterial colonization in that of inanimate surfaces through that of yung ating biofilm. So, for inanimate surfaces, like for example, your nasogastric tube or that of your catheter, or yung mga tubes, di ba, in that of a dialysis patient. Yung mga parang mucoid, parang ganito, na nakadikit dun sa inner surfaces ng mga tubes na yun, those are what you call as the biofilms. Kaya nga minsan, may complication yung mga patients. Like for example, yung mga nagdadialysis na mga patient, they would actually then experience fever, they would actually then experience nausea, they would actually have different kinds of signs and symptoms that are associated with infection because of yung ating biofilm. So once yung blood in certain na mga tubes, like in that of your dialysis tube, so with that of the increasing pressure, that would actually be present dun sa loob ng tube, it can carry yung biofilm at papasok yun dun sa katawan ng tao, and it can actually be manifested with the signs and symptoms of infection. Kaya nga, may mga nagre-reklamo, after nila magpa-dialysis, bigla-bigla silang nagkakaroon ng fever with chills. So that's actually attributed to that of your biofilms. So extracellular polymeric substance associated with your slime layer, it helps cells in a biofilm attached to that of the target environment and that of also to each other para mas maraming colonies yung maprotektahan. And it protects the cells within it. It facilitates communication among them, and it enables the cells to survive by attaching to different types of surfaces in their natural environment. So before going to the different modes of locomotion, any question? May katanungan ba? Guys, may tanong pa ba kayo?
Wala na po, ma'am. Wala po, ma'am.
Gusto niyo bang ituloy na natin to, tapusin na natin to, ay sa mag-extend pa tayo hanggang 1:30? Ano ba yung next class ninyo?
Wala naman na po, ma'am.
Ay, wala na kayo.
Ay, may research po mamaya, ma'am.
Tapusin na lang natin to para ayos kayo makapag-lunch mamaya. So, going to that of the different structures for locomotion, we have your flagella. Zipagan, Mikey Zipagan. Nandiyan ka ba?
Ma'am,
Ano pagkakaiba ng flagella sa pilay?
ma'am, paulit ma'am.
Anong pagkakaiba ng flagella sa pilay?
Ma'am, ang alam ko po, based sa naalala ko, yung flagella po is mahaba po siya and yung pilay po, short po siya, pero thick po siya. Thick streak here din po, ma'am.
Mhm. With regards to locomotion, paano ba ang movement ng isang flagella? Asa na si Zipagan? Hello? Mikey, nandiyan ka pa ba? So, when you say flagella and that of also pilay, with regards to that of its movement, di ba, always nasabing nila whip-like inactivity yung ating flagella, and then hair-like or brush-like movement for that of the pilay. So, for that of the flagella, it's exterior protein filaments that rotate and cause the bacteria to be motile. Complex structures mostly composed of the protein flagellin, and it is a thread-like appendage, very much thick and longer as compared to that of your pilay. And it plays an important role in survival and the ability of the bacteria to cause a disease. It's highly antigenic in the sense that it would actually be contributory to that of the virulence na naman of a particular organism. Like for example, the H antigen present in your salmonella. And some of the immune responses to infection are directed against these organisms. So one particular type of movement that is attributed to that of your flagella is the gliding motility. This is actually seen in your Capnocytophaga, cyanobacteria, at saka myxobacteria. Alam niyo naman kung paano yung gliding motility, di ba? Like, how do you glide ba? When you would actually be training or first time mong mag-skate, di ba? Usually, ang initial, di ba, na steps na pinapagawa nila is to glide dun sa ice, di ba? So that is actually your gliding motility. So flagellum is attached to the bacterial cell body by a complex structure consisting of three different parts. We have the hook, the basal body, and that of also the filament. So the hook, short curved structure that appears to act as the universal joint between the motor and the basal structure and that of the flagellum. So it's actually the one that's responsible not only for attachment but the entire motility of yung ating flagellum. The basal body is the groundwork. It bears a set of rings, one pair in gram-positive and two pairs in gram-negative. Again, take note of the difference between the gram-positive and gram-negative. And then the filament, which is the long outermost region that is actually needed for propelling that of the microorganism. So constant in diameter and contains the globular protein flagellin arranged in several chains that are intertwined and will form a helix around the hollow core. So with that of motility or the ability of the microorganism to move, we actually have different types of motion. So we have what we call as the run or swim. So the bacterium would actually move in one direction for a length of time. If you would actually have observed na how a particular tadpole will actually move, parang ganun yung run or swim. Tuloy-tuloy lang siya, dire-diretso in just one direction. Pero kapag tuloy-tuloy siya, tapos biglang nag-stop, nag-shift, nag-change ng direction, that is runs. Kasi Maraming run at a particular amount of time. Tuloy-tuloy yung movement niya in one direction and then bigla-biglang mag-shift. That's runs. Paano naman kapag tuloy-tuloy yung direction niya and then bigla-bigla siyang magba-bounce back? That is actually your tumbling or tumble motion, di ba? With tumbling, pabaliktad. So it actually causes a reversal of flagellar motion. But the most important for you to remember dito sa topic ng flagella would actually be the different types of your flagella. So, we have atricus with the word meaning none, with the prefix rather, meaning none, so without a flagellum. And then we also have your monotricus, so with the prefix mono, so isa lang yung flagella. One flagellum at one end. And then kapag amphi, that's actually one on both ends. So single flagellum at both ends. And then kapag lophotricus, tough or group or several flagella located on one end or that of both ends. Kapag peritricus, studded talaga siya. Buong katawan niya punong-puno ng flagella. And different types of arrangement nitong ating flagella will then contribute to the motility of the organism. Like, for example, itong monotricus, it would actually be capable of doing yung run or swim. Diba? And then the peritricus, it can actually also not just run, but it can also tumble kasi buong katawan niya punong-puno ng flagella. So next, we go to axial filaments, which are only seen in that of yung corkscrew natin na bacteria, like for example, your spirochete. So, your axial filaments, bundles of fibrils that arise at the end of the cell beneath an outer sheath, and it would spiral. It would actually then coil around that of the cell. So it's anchored at one end of the spirochete. It would actually have a structure similar to that of the flagella, and the rotation of the filament will produce a movement of the outer sheath that propel the spirochete in a spiral motion. Kaya nga, di ba, with its structure, paikot kasi siya, nakapulupot siya. So it would actually give the organism a spiral or corkscrew na motility, yung paikot. So, very important dito yung ating spirochete. Again, never forget yung different types ng ating spirochete. So, this would actually be a group of bacteria that have a unique structure and motility by means of your axial filaments, or in other books, it's regarded as your endoflagella. So, the true motility, and that of also the Brownian movement, they are best observed through a method, eventually madidiscuss yan sa lab, yun yung ating hanging drop. So, when you say true motility, the bacteria would actually be going in a definite direction. So, tuloy-tuloy lang siya. And there would actually be no cuts in that of the motion of the organism. Yung Brownian movement naman, it would actually bounce back and forth once it would actually be able to hit a particular structure. Like, molecules of water. If maalala pa ninyo itong Brownian movement sa inyong chemistry, kapag na-hit niya, for example, ang isang particular structure, like for example, a water molecule, magba-bounce back siya. And then, we have taxis. So, with that of taxis, it's actually either towards or away from a particular stimulus. And if this particular stimulus can actually be light, like your phototaxis, or it can be with the use of chemicals, your chemotaxis. And an example would actually be your antibiotics for that of your chemotaxis. Alam ng microorganism that there would actually be an antibiotic in that of the environment. So it would actually then move away from the area of the antibiotic. And then we have the fimbriae, which is actually a short, fine, hair-like appendage found in gram-negative bacteria, not involved in motility but for attachment to that of solid surfaces. So it will enable the cells to stick to surfaces, including animal tissues, or to form pellicles or biofilms. So again, itong fimbriae together with that of the slime layer, it would actually then contribute to that of the biofilm formation. So notorious human pathogens in which fimbriae assist in disease, we have your Salmonella, your Neisseria gonorrhoeae, which is actually the causative agent for gonorrhea, the sexually transmitted disease. Ito yung commonly tinatawag na tulo sa Tagalog. And then Bordetella pertussis, the causative agent for whooping cough. Ito yung tinatawag din nila sa Tagalog na halak. So that's actually for your fimbriae. And then for the villi, so hair-like proteinaceous structure that would extend from the cell membrane into the external environment. Hair-like microfibrils usually produced by flagellated gram-neck organisms, and they serve as adhesives. If nabasa ninyo kay Jawets, there would actually be minor proteins that are located dun sa tip ng pilay. Ang tawag natin dun sa protein, dun sa tip ni pilay, yun yung adhesive. And si pilay, the bacteria would utilize the pilay, and in moving, uunahin muna yung adhesive. Si adhesive muna yung gagalaw forward, and then it would actually then pull that of the entire pilay, and then eventually magre-retract yung pilay, kukunin niya na yung buong microorganism. So, the movement or the direction of the movement of a particular microorganism with the villi, it will then be determined by that of the adhesives. So, your villi is composed also of structural protein subunits known as your piliens. And because of the piliens together with your adhesives, the villi is highly associated with what we call as the twitching motility, very different from the gliding one seen in your flagella. So, twitching, yun nga, the pilus will then extend, mauuna muna yung pilay, and then, magre-retract siya, kukunin niya na yung entire body ng microorganism. So, it will then result in a short, jerky, intermittent movement. And this is highly characteristic for that of your Pseudomonas aeruginosa, your Neisseria, and certain strains of your Escherichia coli. And there are actually two main classes of your villi. We have your common or ordinary villi and the sex villi. So yung common, it would actually be for adherence to surfaces. So it would actually be needed for virulence. And then the sex villi, Mr. Diaz, nandiyan ka pa ba? Hello?
Ma'am? Hello, ma'am.
Denver, ano yung receptor na ulit ng ating sex villi? Ano na yung receptor ng sex villi na kaninang pinag-usapan lang natin? Hello? Denver, nandiyan ka po ba?
Alam.
So, okay. So, di ba yung sabi natin kanina, ang tawag dito, yung attachment site, very important, most abundant protein in the outer membrane, that is your OMP-A protein, di ba? Siya yung main attachment site ng ating receptor, taga-receive ng ating sex pilus during bacterial conjugation. So, the sex pilus would actually serve as a conduit, parang bridge, for the passage of DNA from the donor bacteria going to that of the recipient bacteria. So it's present only in cells that would actually produce a protein known as your F factor. Kaya nga sinasabi doon, F-mediated bacterial conjugation. So the F-positive cells, initiate conjugation only with an F negative cell. That is to transfer that of the the genetic material from the F positive papunta kay F negative. So it then limits the conjugative process to cells capable of transporting genetic material through the bridge, through the hollow natin na sex pilus. So one particular example would be yung ating streptococci. So fimbriae are the site of the main surface antigen, particularly your M protein. And one of the contributory na naman natin na substance in creating that of your M protein, di ba, would be your lipoteichoic acid. Sabi nga dito, lipoteichoic acid associated with the fimbriae, they would actually then be capable of attaching the Streptococcus pyogenes to that of the epithelial cells of the host. Kaya nga, it actually causes that of your rheumatic fever. And if rheumatic fever would actually then worsen, it actually leads to that of yung rheumatic heart disease. It can also produce that of your infected endocarditis. Kasi the organism tends to spread na all over that of the body. And then we have one unique organism, your Neisseria gonorrhoeae, that is able to make a pili of different antigens. And that is what we call as your antigenic variation. Kapag sinasabing antigenic variation, si Neisseria gonorrhoeae can still attach to that of a cell to which this cell, initially, na-recognize niya na nung una pa lang yung antigen ni Neisseria na nandoon sa kanyang pilay. Pero kahit na-recognize niya na si Neisseria, itong Neisseria na to, pwede pa rin siyang kumapit kasi it changes that of the antigen present in that of its pilay. And again, that is your antigenic variation. So all in all, your pili, similar to that of your capsule, would actually then inhibit the phagocytosis that is seen in your WBCs, like for example, your neutrophils and your macrophages. So that's it for your module 2. Any questions? Any questions?